Good morning. Today we'll be starting with Mendelian genetics and its extension that falls under the Unit 1 of your CC12 topic. So these are the references that have gone through while dealing with this topic. So people can go through these references. And given your syllabus, I have indexed the chapter into 11 sections. Even though sex limited and sex influenced is not a part of your syllabus, but I thought that while dealing with all these allelic forms, why not touch this part also? And now we come to the main topic. So long after Mendel in 1953, the discovery of this structure actually ushered the age of molecular biology. And once this structure was discovered, specifically the structure of DNA in the very famous paper of Watson and Crick in 1953, it opened a variety of biological research areas where once the DNA structure was totally known, the concept of gene became very easily available to us. And the central dogma of molecular biology was laid on the foundation that a segment of DNA capable of making a product is a gene. The product can be an RNA or can be any form of RNA. However, if the product is an mRNA which is capable of making another product, that is protein, then these kinds of segments are known as structural genes. While if the product is only RNA, for example, ribosomal RNA, transfer RNA, so then since we are not getting another third product, so those kinds of DNA segments which produce ribosomal RNA or transfer RNA came to be known as functional genes. So this was the concept that developed with the discovery of this structure. But going back, if we move back almost 60 years back, so coming to the era of 1860, when it was the age of Mendel, and it was during this time that nothing was known about DNA. So the basic concept of gene as we know today was not known then. It was in this background that Mendel's work became very important, even though when Mendel did his work, it was around the time of 1856 to 1863, nobody could realize the importance of his work unless and until there were three scientists who later discovered the Mendel's work. I guess you know the name of those three famous scientists. They were botanists Hugo de Vries, Erich von Zermatt and Karl Correns. They actually rediscovered Mendelism, and long before Mendel had died, Mendel passed away in 1884 at the age of 61. So long after Mendel died, Mendel didn't realize in his lifetime that how much he actually contributed to genetics. But long after his death, his work, which was rediscovered by these three famous botanists, actually created an uproar in the area of genetics. And Mendel's work, followed by some of the very famous work of Bridges and Morgan, gradually built up the principle of inheritance. So if Mendel's work was published around the era of, as I said, his lecture was published in 1866, while he worked, his actual experimental tenure was from 1856 to 1863. He gave a lecture on his work in 1866. That was when his work was published. But in 1902, it was around 1902 that his work was rediscovered, and then there was a huge amount of experiments related to the chromosomal theory of inheritance and transmission of genetics. Along with Mendel's work came very famous works of Bridges Morgan with Drosophila, Stuyvesant with Drosophila, and these works together form the chromosomal theory of inheritance. It was around this era, that is 1928 onwards, Griffith started working with DNA, and followed by the famous experiments of Hershey and Chase, and finally the structure of DNA was discovered around this era. So unless and until the structure of DNA was discovered, the concept of gene at the biochemical level, at the molecular level, could not be elucidated very properly prior to that. But everybody has had hypothesized that there is something that is being inherited, and this inheritance follows a definite pattern. Now comes the era. So once this famous work of DNA was discovered by Watson and Crick, and we know the model of Watson and Crick, then developed the era of recombinant DNA technology, DNA cloning, and finally, as we moved on to this century, it was the era of Human Genome Project, genome-wide acetylation studies. The era of epigenetics started. So the application of genomics started. So what we will be actually discussing in this unit 1 of your syllabus is going behind a bit to the era of Mendel. As I said that it was in 1900 that Mendel was rediscovered. Not only that, there was another famous scientist, you have heard his name or not, I'm not aware, but his name was Archibald Garrod, who successfully applied to the inheritance of alkaptonuria, which was then becoming a widely known subject among biologists. However, the principle on which Garrod based his hypothesis was somewhat based on the hypothesis of a scientist, Johann Gregor Mendel, who his life was from 1822 to 1884. Mendel was born in Czech Republic. Although his parents were simple farmers with little money, but he could achieve sound education and was admitted to the Augustinian monastery in Brno in September 1843. From therein, he went on to study in the University of Vienna, which he attended from 1851 to 1853. After 2 years of study in Vienna, he came back to Brno, where he taught in a school and began his experimental work at the backyard. He conducted breeding experiments from, as I said, 1856 to 1863 and presented his results publicly at a meeting of the Brno National Science Society in 1865. And Mendel's lecture was published, as I said, during this time in 1866. However, in 1868, Mendel was elected as the abbot of his monastery, and the increasing administrative duties almost brought a halt to his teaching and scientific career. He died at the age of 61 in 1884, unrecognized for his contribution in genetics. There are very few scientists in the world who get recognized for the work they do in their lifetime. Watson was the luckiest of that lot because he got recognized for his work at the mere age of 25. And here we hear the story of Mendel, who when died, he didn't know that his work of Pisum sativum would be one of the seminal works of genetics based on which the whole foundation of genetics would be laid upon. And as I again said that later, Mendel's work was rediscovered almost after 1884 he passed away. So it was almost after 18 years after his death, his work was rediscovered, and then thus thereafter Mendel came to be known as the father of genetics. So this is Gregor Johann Mendel, and this was what briefly I told about the history of Mendel. And this is a photo of Mendel experimenting with the peas. And now coming to this concept. When we think about genetics, there are two factors or two words which you have on and off heard. One is the genotype, the other is the phenotype. Genotype means the genetic constitution of an organism, whatever it is written in the genes. Phenotype means that whatever is written in the genes, the physical expression of those things written in the genes. So is the physical expression of a physical trait. However, there is another factor that we have to take into consideration, as Darwin in his evolution theory very eloquently proposed that nature plays a role. So environmental influences and random developmental events. So obviously there is a role of nature. So together the environmental influences and the random genetic developments along with the genotype of the organism is what actually makes the phenotypic expression of an organism. These are some important genetic terms which is important when we think about in terms of monohybrid cross or a dihybrid cross. Gene, now we know, is a segment of DNA, but when we are thinking of Mendel, it's a genetic factor that determines the characteristics. Allele. Allele is one or two more alternate forms of a gene. Let me just give an example of Mendelism. You have heard about the character length because you have studied in your earlier classes that the pea plant can exhibit two categories of height, the tall and the dwarf. So the character here is length. Okay, now the length character can have a phenotypic expression of a tall plant, or the length character can have a phenotypic expression of a dwarf plant. If the tall character is represented by capital T, with respect to capital T, the dwarf character is represented by small t. So, if the character, so the gene is that determines the characteristics. So, the characteristic is the characteristic of height. Height can exist as capital T or tall variant and as small t in a homozygous form or a dwarf variant. So capital T and small t becomes the allele for the character length. Okay. Locus. Locus is a specified place on a chromosome occupied by an allele. So where the gene is sitting, that's the locus. Plural, we say loci. So the plural is loci, singular is locus. Genotype, set of alleles possessed by an individual organism. So, for example, if we think of again a tall plant can be represented by capital T, capital T. This is something I am recapitulating from what you have studied in 11-12. Capital T is a tall plant. Capital T, small t is also a tall plant. So, these are genotypes. Okay. Again, small t is a dwarf plant. So that is also, so small t is a dwarf plant, and we are getting that is the genotype. Phenotypically also we are seeing that it is a dwarf plant. Capital T is the genotype. Phenotypically we are seeing that it is a tall plant. Okay, so that's the genotype. Heterozygote. An individual organism possessing two different alleles at a locus. Again the same example. So if it's a capital T, small t, that's a tall plant. So the character is that of height. The height of the plant is tall. That is phenotypically we are seeing a tall plant. But when we think of the genotype of the plant, it's a capital T in one locus, and just on the opposite side on the other chromosome a small t is sitting. So that's the heterozygote. Homozygote is when you have the individual organism possessing the two same categories of allele. So it's capital T or small t. Phenotype or trait, the appearance or manifestation of a character, how you see a character, how it is being expressed. And the last is character, an attribute or feature. So the character is an attribute or feature that is being observed, that is under study. So when we think of Mendelism, so what were the characters he considered during his experiment? He examined 7 characteristics. So it was the seed color, seed shape, seed coat, flower position, stem length, so it's tall and short. So tall can be represented as capital T, homozygote, or capital T, small t, heterozygote, and short is always small t. This is axial, as I said, along the stem or terminal at the tip of the stem. Pod shape, how the pod looks, yellow and green. So the pod color. So he examined these 7 characteristics. There was a luck favoring in case of Mendel when he chose these 7 characteristics. The luck that favored Mendel was that these characteristics are present in only 2 distinct forms. So either you have a yellow pod or you have a green pod. You do not have an intermediate something. So that was one of the luck that Mendel, when the Mendel chose the characters, he avoided characteristics that display a range of variations. So he just avoided those characteristics that displayed a range of variations. That was one thing. And he focused his attention on those that exist in 2 easily differentiated forms, as you can see. White versus grey, round versus wrinkled, yellow versus green, inflated versus constricted. So these were 2 distinct forms. There was nothing intermediate. So this was one thing where his luck favored. Second was the model he chose. That was the pea plant. Compared with certain other plants, pea plants grow relatively rapidly, completing an entire generation in a single growing season. Later on, in the animal model, Drosophila was chosen for this property because we get generations of Drosophila very quickly. So as I said, there are 2 things. There was a luck factor that favored Mendel when he studied his work, the choice of the plant and the choice of the characters. Finally, there's another thing that helped Mendel to achieve such success was that he was very meticulous in documenting his results. And unlike other investigators who worked in this similar area before him, he just described the results by formulating hypotheses. So he had a hypothesis based on his initial observations, and then he conducted crosses to test his hypothesis. So there was a statistical component to his experiment. Any experiment that has a statistical support is a very foolproof category of experiments. Not only that, he was very meticulous. He kept careful records of the number of progeny possessing each type of trait. So he just didn't keep it in his mind. He observed, he wrote the data methodically in his diaries. That is why in 1900, when the 3 botanists started working separately, and how could the work be compared to Mendel? Because Mendel's work were very methodically jotted down in his diaries. And so as I was saying, so he kept careful records of each type of trait and computed ratios of the different types. So as you can see, he worked for almost 10 years, and for this 10 years he was very meticulous and thorough. That was his scientific component, but the luck factor was that the choice of his traits and the choice of the plant. So that was his luck factor. And these, as you can see, are the results he obtained. So as I said, there were 7 characters he took into consideration, and you can see so from the crosses he observed, so you can see the amount of data he observed. So it was 5474, you can see the count figure, 185474 round, 1850 wrinkle. So anyhow, I'm not, just a view of this data gives you that with how many I mean to say, what was the number with which he dealt his crosses with. When you're thinking of a plant, you can see he did the crosses with 787 plants, 277 dwarfs. So you can estimate the number of plants it took to do his cross. So his sample was, for a human to do it all alone in your backyard, was quite huge. So whatever the crosses he did, there was an interesting result was that he observed a ratio of 3:1. And that forms the famous, the first, I mean to say, when we talk about the monohybrid cross, that is his famous result of this 3:1 ratio. And initially, so there are two laws of Mendel you should keep in mind. One is the law of segregation, and the second was the law of independent assortment. In the law of segregation, he talks about, what does he talk about? That each individual organism possesses two alleles encoding a trait. So if we think about this example, so each individual organism, for example, if it is a tall plant, the tall plant is possessing two alleles which defines its trait of being a tall. In this case, the tall plant is having the two alleles, capital T, one allele, small t, the other allele, if it is a heterozygote, and a capital T, one allele, and the small t, another allele, if it is a homozygote. So that was his first hypothesis. This is the first point of his observation under the law of segregation. The second observation was that alleles separate when gametes are formed. That's another thing. So obviously, I'll be coming to that picture later on. So when gametes are formed, the alleles separate during anaphase. So it is in the meiosis stage of anaphase that the alleles separate and in equal proportions. So now let's come to this writing part. So a plant can be a homozygote, Y by Y. That's a dominant trait, that is a recessive trait. This is a homozygous recessive, and this is a heterozygous. How are alleles written? As you can see, a capital Y / capital Y. So this is basically this is how the alleles are written, and depending on if it's a dominant trait, then we write it as capital Y, and with respect to capital Y, the small y is written. Now, if the gene is phonetically called Y, it sounds Y, W-Y-E, when you pronounce it, it sounds Y. So if it's phonetically called Y, then we represent it in this manner. Interestingly, when we think of these three kinds of allele, as you can see, there is a letter alphabet written in capital and an alphabet written in small. The alphabet written in capital is dominant and the alphabet written in small is recessive. And the first hypothesis that came from Mendel was the Mendel's law of equal segregation. That is, as I just now said, each individual organism possesses two alleles encoding a trait. Alleles separate when gametes are formed. And when we think of the different combinations, a homozygous dominant is Y / Y, a heterozygous dominant is capital Y / small y, and a homozygous dominant again is small y / small y. In genetics, generally, allelic combinations underlying phenotypes are called genotypes. So, phenotypically, we are getting two varieties. This is tall and this is dwarf. Okay. Genotypically, we are getting three varieties. This is a homozygote tall, this is a homozygous dwarf, and this is a, this is a homozygous tall, this is a heterozygous tall, and this is a homozygous dwarf. And now coming to that experiment part, just as I said, gene exists in different versions called alleles. So, if this is a position where a gene is sitting, now we know what a gene is. So we have allele, there are two. So this allele obviously codes for a character. So now let us assume that it codes for the character round seeds. So that's the dominant character, then smaller represents the wrinkled seeds. So the character is round or wrinkled seeds. The two alternative alleles are capital R or small r. The two alternative alleles are capital R or small r, and they are occupying the same locus in the two sets of different homologous chromosomes. Okay, so this is in brief the Mendel's experiment. So when he did crosses with the pea plants, he ensured that there should not be any self-fertilization. So he removed the anthers from the female flower and brushed dusted the stigma with the pollens from a different flower. Okay, so you remove the anthers and dusted the stigma with the pollens from a different flower, and the pollen fertilized ova developed into seeds, and then he got round seeds and wrinkled seeds. Initially he got all round seeds. Anyway, so he got round and wrinkled seeds. That was a P generation. And on crosses, this is the first filial generation, all the seeds were round. Next, when he self-fertilized these round seeds, he got 3/4 round and 1/4 wrinkled. So, the first conclusion he drew was that the traits of the parents do not blend. We started with a round seed and a wrinkled seed, and finally when we got the ratio of 3:1, we get a round seed which is one of the parent and a wrinkled seed which is the other parent. So the traits of the parent do not blend. Although the F1 display the phenotype of one parent, both traits are passed to F2 progeny. This is interesting. So in the parental generations, when this cross was done, what was observed that all the F1 plants have round seed. Okay, so we do not see this trait of wrinkled seed in the F1 generation. But interestingly, this trait of wrinkled seed gets expressed in the F2 generation. So in the F2 generation, both the traits of the parents have passed. So obviously there must be something about this round allele and the wrinkled allele, and thus came in the concept of dominance and recessive. So this was Mendel's monohybrid cross, and the monohybrid cross actually revealed the principle of segregation. So now let's look at it into a bit detail. So as I said, these were the round seeds. Let us assume it's a homozygous round seed. He took a pure breeding one with a homozygous wrinkled seed, and as you can see, the gametes are formed. So the two alleles in each plant separated when the gametes are formed. So each gamete, so there will be two gametes from this one, capital R, two gametes from this one, small r, small r. Fertilization occurs, we are getting round seeds. Now we are self-fertilizing. So each round seeds will again present two gametes, capital R bearing one gamete and small r bearing one gamete. And now they will be self-fertilized. So when they will be self-fertilized, what we observed in the earlier slide was that there were round seeds and wrinkled seeds. That was a phenotypic ratio. Now let's look at the genotype. So we are getting pure round seeds with two RR gametes. This will produce capital R gametes. Heterozygote round seeds which will produce two variant of gametes. One set of gamete will be a capital R, and the other set of gamete will be a small r. And the wrinkled seed will again produce gametes which will be a small r. So this was the F2 generation. Now he also self-fertilized the F3 generation, and again you can see we are getting the same ratio. That's 1/4 pure round seeds, as you can see. So to produce and then we are getting pure round seeds, then we are getting heterozygote round seeds, and we are getting wrinkled seeds. So this parent will always, if a wrinkled cross with a wrinkled, pure breed will always produce a wrinkled seed. If a round, pure breed round crossed with a pure breed parent will always produce round seed. But when we do this kind of crosses, a heterozygote round crossed with a heterozygote round, we will always obtain the ratio of 3:1, where 3/4 of the plants are round phenotypically and 1/4 of the plant is wrinkled phenotypically as well as genotypically. So he established the principle of segregation, that each individual organism produces two alleles encoding a trait, Alleles separate, as you can see, the alleles are separating when gametes are forming, and alleles separate in equal proportions. So this was his principle of segregation and the concept of dominance.